last speaker just before lunch is Vincenzo Vitelli, a professor here at the University of Chicago, also in the physics department, and is going to take us more to the soft and low energy end of the physics spectrum. Thank you.
Before my slides come up, let me first say that I'm very happy to be here, and I'm grateful to the organizer for the opportunity to speak and tell you more about learning simple models from biological data. And if the screen cooperates, I'll tell you what the biological data is. You can see here this image. These are not particles. They are cells moving, flowing on the curved surface of an embryo. What you're seeing here is a clip that documents the first 20 minutes. Oh, yeah. That describes the first 20 minutes in the life of a fly. And the fly is a model organism, but this phenomenon, this process is universal, it's called morphogenesis. You and I come from something similar to this. And here you see, if you like, one single cell on a substrate, itself in a machine of excruciating complexity. And what we would like to do is to build physical models that describe this stuff. So perhaps the first question to ask is, what is a physical model? And I would like to start by asking myself, what is the method of theoretical physics? The method in which I was trained. It's a subject of great beauty, built on minimalism. More clearly, it says that once you know the symmetries and the conservation laws that apply to a given physical situation, you can immediately determine the collective variables of the field that describe that phenomenon. And those fields and variables must satisfy some dynamical equations, the form of which is highly constrained, aside probably from the parameters or the constant that enter those equations. This enormous simplification is the basis of the success of physics. A lot of great examples you heard in the last three talks of this. We have Hamiltonians, actions, from which the dynamics can be derived. The problem of this approach is that it doesn't work if what you want to describe is alive. It only works for dead things like beats, masses, springs, or maybe the first 3 minutes in the history of the universe. But if you want to understand the fly, yeah, that doesn't work. And why doesn't it work? Well, think about conservation of energy. Like, I don't conserve energy. If I'm tired, I get a cup of coffee and keep going. Similarly, the cell. So if you go beyond the cartoon of a cell, and you just look, okay, how does it look? This is a cell. It doesn't have a rest date. It keeps remodeling constantly. Sure, this is on a substrate. It will exercise forces on a substrate, but I'm hard-pressed to call this mechanics. And what's most crucial about it is that the program that this machine is following is encoded in microscopic information in proteins and genes. And this information propagates across scales to describe the mechanics of the physics of this organism. And so the central problem here is to find even a primitive way of making inferences starting from biochemistry about the physical world. And this is the two-way channel. Okay? And this is an exercise for which machine learning techniques are eminently suitable. What we would like to do, our dream of restoring what we lost, is to channel that information in some pipelines that eventually gives us equations. And not necessarily because equations are better at predicting things. In fact, often neural networks are just as accurate, if not better. But once we have those equations in front of our eyes, we can just look at them as we always look at them to see mechanisms that explain physical phenomena. And if we can understand or learn, maybe it would be more correct, this physical phenomena, we can ask our experimental friends to test them by applying the wealth of biochemical perturbation and mutant analysis that biology gives us. I'd like to just give you two examples of doing that, working really close with our experimental colleagues. And the first problem that I would tell you is that how does that cell that you show generates forces on a substrate? This is a mechanics problem, seemingly easy to define. And none of this work would be possible without our experimental collaborators, in particular Margaret Gardell here in Chicago, Patrick Oaks of Loyola, and their top postdocs, Stefano, John, and Sharaya, who was a Schmidt Fellow. And I also like to acknowledge my PhD student, extremely talented Matt Schmidt and Jonathan Cohen, were responsible for all the modeling and AI work. So let me tell you what we're doing. First of all, the problem is not easy, but Margaret and Patrick are pioneers in a technique to measure these forces. So we have access to the forces that the cell, whose perimeter you see here, generates on the substrate. This is a map, and the intensity of this color map denotes how strong those forces are, highly homogeneously distributed. What's even more useful for our purposes is that what Margaret and Patrick can do is to also image fluorescent, you can generate fluorescent images of proteins. All the proteins that deem to be implicated in creating this mechanical response. So our first goal, the least ambitious one, is can we build a protein fourth mind? What is that? If you've never heard of it, don't worry, it doesn't exist. And in fact, even if you can build it, it will be an in silico device. A neural net that given images like this of proteins tells you what the forces are without going in the lab and measure. This is a tool to be useful enough, especially if you want to perturb a protein distribution and anticipate the consequences for bioengineering. But that would be only the first step that we're going to use towards building what I would call a canonical model. So the first thing that we need to understand is that what are the hypotheses around about what generates these forces? And one school of thought that I understand is not the only one in biology is that there are special places where the cell adheres to the substrate that are responsible for generating those forces. The subject of adhesion is very complicated. You know, usually physics, theoretical physics, does very poorly when confronted with the ordinary. So if you want to understand how two pieces of two sheets with a little bit of glue in between slide on top of each other, this is a very challenging problem. It's very easy to do, but it's very challenging. Even more challenging here. Now, because physics is typically going with assumption, simplicity, it's almost unnerving. You imagine that an addition site is a point where there's a spring that sticks between these two cells. Now you go to the chemist and you ask, okay, can you give us a simplified picture of how a focal addition site looks? And they'll come up with this. And this is a simplified picture in the sense that what they're listing with this diagrams is a list of proteins. All of them, there's solid evidence biochemically. They play a role in describing and determining the forces at the level of cause and effect. But there's a lot of them with all their exotic names. And in particular, some of them are spread throughout the cell. Some of them are localized in this focal addition site. So the first question that we wanted to ask is, does an effective mathematical model require all of these proteins, all of these fields? We like to reduce, if you like, the dimensionality of this problem. Please notice, as a preemptive announcement, I'm not implying that only one or two of these proteins are needed. The statement we are trying to construct is what's the minimal amount of information needed to construct a model. And so the first step we're going to do is to put together physics and chemistry. And we'll do so by first using machine learning techniques to identify the proteins that are predictive of the forces, essentially by trial and errors. And then we're going to use that information to build a chemophysical model. Okay? The first step is the protein force microscope. So what we've done is we've taken lots of images, thousands of images of fluorescent proteins, all of the ones that their experimental friends suspect are implicated in this process. And then we trained a neural net using experimental images of the forces to predict, on a new batch of data has never been seen, what are the traction force maps. As you can see by comparing these two images, this is a pretty stunning agreement. Now, the thing that surprised me is that I was prepared for a mess. I was prepared that I would have needed multiple channels simultaneously. But this type of accuracy, which is really good for me, was obtained using only a single protein. Now, I want to stress that doesn't mean that that is the only protein biochemically responsible. It means that there are correlations among all of these proteins. So we could hope to construct a single master field to be coupled with the physical equations that describes the physics of this phenomenon. Okay? Now, of course, the way we did it thousands of images split in train and test data. And here is the angle that these forces do in our neural net prediction. The experiment, straight line here is perfect prediction. There's a bit of spread. It's a bit harder to predict the magnitude of the forces, but we're not doing badly either. Now, of course, you could repeat this exercise for all those protons, and including also some negative control, things like mitochondria that we know have nothing to do with forces, or just the mask of the cell, and measure the errors that you get in your neural network performance. And using the magnitude of the forces, which is more sensitive, what you can see here that there are some proteins that do better than others. Zixin in particular, but also this one called paxillin. The commonality between these proteins, these are focal adhesion site proteins. They only found in those places, corroborating an hypothesis, at least at this early stage. What's interesting also is that if you try to put two channels, say Zixin plus some of these other proteins, you don't improve significantly the prediction. So given that the accuracy was already super high for our taste, we're going to take the advantage of having only one additional field and try to go with it and build models. But not before testing the robustness of our machine learning model to perturbations. So this is one of the beautiful things you can do with biology. So Margaret and Patrick and their gang can go in and knock off, work out, actually this is the technical word, work out one of the protein mice. It's not Zixin, it's another one that really controls the mechanics of cells. And then what we can do is we can use our machine learning protocol that was never trained on this cell with a washout to see how well it does in prediction. In particular, here there is the total force versus time. The dashed line is machine learning. The blue one is machine learning. The gray one is our experiment. This is the point where the perturbation is applied. And here you will see movies that help you to correlate it. And the part I would like to see you that these curves are on top of each other throughout time, even if the cell was never seen before. So our neural network can generalize to perturbation that it has never seen before, which is the first step towards start taking its conclusion, at least as an hypothesis, to be verified. If we want to do better than that, we have to find a rational way of connecting biochemistry to physics, using perhaps the fact that we just learned that Zixin is an excellent predictor of forces. So here we have to go back to physics. And this is a state-of-the-art model in this field from 2014, also involving some of our own collaborators. This is experimental image. This is how this model does. Certainly not a remarkable success. But the physics in this model is, if you like, as complicated as you can create using purely physics reasoning. The first term, I won't define everything, is just the elasticity of your medium. The second term is an active contractility of this network. And the third one is an adhesion, which literally treats the cell as if it was a sheet glued on a substrate with some strength Y. Okay. And this model is entirely homogeneous, doesn't know about focal adhesion site, okay, because that's not physics information. So one simple thing that we can attempt is if the focal adhesion site are responsible, maybe there is a way of promoting this parameter to a field with its own dynamics potentially, and enhance continuous elasticity with what we learned from machine learning, making this field position dependent. But the question is, how do you determine that? Well, what we're going to do is that we're just going to learn it using Zixin, because you know that Zixin is predictive of those forces. So what we're going to do is just tweak a little bit our normal architecture, and which starts with fluorescent images of protein and produces forces. We're going to stick in between what we call a physical bottleneck. We're going to ask the neural net to predict the parameters of this model, and then use a PD solver to predict the forces. And then we are going to be guaranteed that this updated group respects physics, while being able to learn this problem, this field, which is unknown to us. And so this physical bottleneck approach allows us to do prediction that are still in very good agreement. It allows us to extract this adhesion field, and it continues to behave well, even when we apply biochemical perturbation like this are often needed. And one question you can ask is that if you want to predict this adhesion field, how far out you have to absorb information from this? This is like trying to extract a length scale. And the answer here is a few microns, which is the characteristic length scale that is deemed to be associated with this focal emission site, corroborating again our hypothesis. So what I would like to say that this first step kit has shown the following. Now, of course, I mean, our goal is to describe experiments and potentially explain them. And now the first thing that you can do in a pipeline is an imaginary graph, in which there's description and simplicity measured by some arbitrary units. And the neural net, in my opinion, doesn't fire particularly well in terms of insights. I mean, it can tell you what are the relevant variables, but it is extremely good in terms of descriptiveness of the data. The other thing you could do is to march from the other side, take a continuum model, enhance it with this physical bottleneck neural nets. And here you'll have all the advantages of interpretability, but also an enhanced power of descriptiveness. It's something I'll have no time to describe, is that there's something in between that we call Green's function neural network, which you don't assume exactly what physics you have. You just assume that there are some generators and Green's functions, and you learn both the Green functions and the field on the fly. That lies a little bit in between. Now, in this example, the protein, we didn't write an explicit dynamics. I want to show you an example in which we can do really a standard model, a closed loop dynamics between physics and biochemistry. And for that, I want to resort to this image. So again, this is the top of the fruit fly embryo from the Stryken lab at Santa Barbara. And what you're going to see here initially is a fluid of these particles that are celled moving on a closed substrate. But gradually, this fluid and the underlying substrate will turn into a lava. This is the process of morphogenesis. We want to understand the first 20 minutes of this process, where you go from the cigar-shaped object to setting up the body of the organism. In parallel to what you can see under the microscope, there is the pattern of genes that underlies and powers that dynamics. In a moment of Germanic lyrism, my experimental collaborator, Sebastian Stryken, called it a system of coordinates for cell fate, because it literally planned in it is how the cells are going to move and form the body of the organism. What we want to do, not surprising perhaps, we want to go from this to that and create a mathematical model that predicts that until at least this cigar is roughly cigar, this moment in which the tail starts falling. Okay? And of course, this is only possible because from the immense help from Sebastian's lab, Nicholas, Fritjof, Matt, Noah, Marion, and of course, Jonathan Poland, who's an extraordinary student responsible for all the machine learning results here. And this is some unpublished result. So the fly is our target. Okay? There are two things biochemically that you can think determines the fate of that fly. One is gene patterns, and the other is proteins. But the two are not decoupled. They talk to each other. And one of the questions we want to answer is that which of this information do we need to put to do a closed loop dynamical model for this system? All of it is important biochemically. And just in terms of language, we're going to use the language of, we're going to define, you notice here there's a beautiful pattern formation problem. So we are going to define the direction from anterior posteriors of the organism, the AP axis, and then the dorsoventral is going to be the perpendicular one. And notice that there are beautiful gradients in the distribution of this protein. And we want to understand what is the mechanism that will lead to this morphogenetic flow, the fluid mechanics problem that you saw. And of course, you can unwrap, as we'll do for the simplicity of viewing, this morphogenetic flow in a projection. And you can see classic signature of vortices. But these are not the vortices that are determined purely by Navier-Stokes equation. Okay, because Navier-Stokes equation doesn't know about protein genes. Okay, you have to build on it. Right. Okay, so how are we going to do that? First step, as before, identify the minimal variable, or at least formulate hypotheses for what they could be. This is what we're going to do with neural nets. Okay, and then we're going to extract them out. So let me jump at the thing that got us a little bit interested in this business. This is Jonathan taking only 2 proteins at the beginning of time and predicting 20 minutes in the future from here when nothing was moving to the tail start forming using only 2 images of 2 proteins. This is the experimental data plotted out for the flow. This is the machine learning result. I mean, I can show you metrics, but you can also just overlap and see. Okay. So when we saw that, we can say, well, how did you pick up those proteins? Same idea as before. we have in principle many patterns of genes, combination of proteins. We picked up the two ones that minimize the error in predicting, and they are myosin and cadherin. As it happened, we also have experimental data for all of these proteins as a function of time. Okay. What we want to do now, we want to write down a closed system of equation for the density of cadherin, the density of myosin, and the flow. Physics married to biochemistry in a single predictive framework that we can test and interpret. That's the goal. So what is known? What's known at the moment, before we start working, is little, mostly due to our collaborators, as a matter of fact. First of all, this is myosin, and the first thing you can see, for the expert of treat problems in the audience, you'll be horrified to know that we're going to treat this as a continuous variable, despite there's an obvious graph showing there. And the density of this continuous variable is clearly graded. There are gradients in the dorsoventral direction. It's yellower here than up there. This is the myosin protein. And the standard picture is that the whole flow on top of this embryo can be understood as follows. There's an equation, which is a physics equation, which is Stokes' equation. And then there's a source, which is gradient of the myosin. Myosin is a tensor. The divergence of a tensor gives you a vector field. And that's the whole logic for writing down that as a simplest term. Okay. The problem with this is that you want to understand how the gradients in myosin are built in, otherwise there would be no flow. What regulates them? And as the density of myosin changes in time, how do we close the equation? This question out there. We need mathematical closure and the additional field that allows us to build it. This is what we're going to do. But once you do have that, this gradient will allow you to, just from, if there is a gradient in this direction, there will be a force in this direction. The cell will want to move like that. The conservation of mass, they have to go somewhere. They go this way and that way. That is what generates ultimately the flow that we have. So the central problem is what sets those gradients of myosin, what process, what mechanism, and then can we elucidate? So the first thing is that these tissue flows are simple. Simplicity is always in the eye of the beholder. But they're not simple in the sense that I can use conformal math and solve for them. But they're simple in the sense that if I do a principal component analysis, only a handful of components are enough to describe that flow at any given time with relative weight that changes the function of time. And here I just plotted the flow of the system in a space of two components as a function of time. Notice that every embryo has a somewhat different trajectory revealed in parallel, but there is an ensemble mean that we can use to try to elucidate the behavior of the system. And we can actually look at that. For example, at the early time, what we call the zero, when the flow just starts, this is the experimental picture of that flow, and is well absorbed by a couple of components. If you look at later time, here you can identify the same feature. If you look at later times here, it's even more obvious because this is the flow field experimentally, and it's almost entirely component one. Okay. So we have essentially a latent space, which has a reduced dimensionality. And we can do the same game also for the proteins, cadherin and myosin. And again, the principal component, the one is most important, describes, and this is a very important hint, gradient in the dorsoventral direction, or in the spatial distribution of this protein. And here, space is always important because the flow is in a curved space, and the proteins are non-uniformly distributed in a way that we want to understand. So now, what we would like to do is we would like to connect the dynamics in that reduced space for the flow to the dynamics in this reduced space for the protein. Now we're going to do that. Well, if we can do that, we can build a model. So we're going to do it as follows. It's a very poor man's version. You take the protein fields and you build a library that composes hundreds of terms. If this was standard physics, we could say which term should be there based on symmetries and conservation laws. We don't have that luxury, but we can certainly guess unlimited imagination, limited only by our own computers. The next step is to project, if you like, those each term of this library on our principal components. And then the third step is to do sparse regression to determine their dynamics. And I'm going to show you the outcome of that analysis. There's going to be some term that we know exists, like convective derivative. Every fluid will have that. So we don't have to learn those, so we impose them. But there's going to be other things. Okay, one is obvious. I mean, some of this myosin can just abandon the junctions and be destroyed. But things that are particularly important will be mechanochemical feedbacks. Like the junction between cells can be stretched by the flow. And that flow recruits more mice in a way that is proportional to how much bacteria you have. This is what we wrote succinctly in this term. This is a term that, I mean, you could have hypothesized, but you could have never understood just from movies that this is the key one determining the flow. And there are others that, in the interest of time, I will not go through. But this one I single it out because it's crucial for the mechanism. In a sense, I'm going to clarify momentarily. The bottom line is that if we take these three equations, we have our standard model. It's a closed set of equations. It's not a standard model of elementary particles. It's only a standard model that describes 20 minutes in the life of a fly. Okay, this is what we spend our life on. Now, of course, I should say as a disclaimer that there are commonality across species. So if you understand the fly, maybe you can understand you myself. But of course, it's a long shot. Now, like any model, you don't only need equation, you also need the initial condition. And this is where this term is particularly important. The initial condition at time equals 0, which is known as ventral furrow, when nothing moves. For myosin is flat. There's no gradient in myosin. So the thing that we need to power the flow is absent. This gradient has to be generated. But theory does have a gradient in the dorsoventral direction. This term is the mechanism that couples the mechanics of myosin to the distribution of cadherin. If you like, cadherin is an inhibitor for that mechanochemical feedback that establishes the gradient in the dorsoventral direction. Once those gradients are there, flow ensures and development starts. So what we can do now is that we have an autonomous system of dynamical equation. We have a set of initial conditions. It's like any other model, these equations, we can just integrate them, make plots of myosin, cadherin, and flow, and compare them with experiments. They're in excellent agreement. But again, you remember what I really care is not necessarily agreement and predictive power, is the mechanism. So here is the unique ability offered by working side by side with our experimental collaborators. We have identified the mechanism that makes this work as establishing these gradients of a protein in the dorsoventral direction. This is a very specific mechanism. What the experimentalists can do is that they have the silver bullet. They can make mutants that do not generate those gradients. So our model has to break down whenever you have a mutant that does not have those gradients, and development should not have it. So this is these two mutants. You can sort of see here, this is a bit less visible, but here there's a uniform distribution of myosin at all time, because this mutant is unable to create that gradient. And this mutant have exalted name, Spetzel and TOLRM9. And when you try to see the dynamics generated by this mutant, even just by eye, this embryo is no longer undergoing development. The vortex structure that powers development is entirely suppressed. Our model doesn't work, but development doesn't work either. This validates the mechanism, even at a qualitative level. And that is the type of things that we really like to do more and more. So I hope that I convinced you that there are pipelines that substitute rigorous mathematical thinking or supplemented that allow you to go from experiment all the way to models by just, if you like, infusing the standard techniques with data-driven approaches. Thank you very much for your attention.
Yeah, really fascinating. I think it's following up on what we were talking yesterday, right? So you talked about the Green's function approximation. I guess one question is that, I know that's the linear model. So is that assumption a good one in this case?
Yeah, so in this case, we could write down with a high number of field linear approximate. But after we developed the model, we realized that the name was not very good, because Green's function neural net immediately indicates Green's function notionally applies to linear problem. But whenever the first thing we did, I asked, can you please test on something nonlinear, like for example, shock propagation, the Berger's equation. This is a nonlinear equation, although it's known that there is a mapping, a nonlinear change of variable. These techniques continue to work even in that case. So perhaps it's a bit of a misnomer.
Got it.
So,
So you're thinking as it more as a Koopman kind of
Exactly,
thing where,
exactly.
you know, you're lifting it.
Yeah.
Right. Thank you.
Thank you for the talk. When you're using these neural networks to like, determine what the minimal variables you need for this predictive model are, how do you know when to stop? Like, how do you know when to trust your results or like, you know, not attribute it, like the like poor performance of the results to like, you know, just setting up the machine learning suboptimally?
So we have a great advantage in this business, is that we compete with zero. And literally, I mean, we have to construct this equation from zero. So, so the first step is you decide arbitrarily what is good accuracy that you would like to settle for. For me, qualitative agreement is good enough if I can interpret it. But of course, you can see that was way beyond qualitative agreement to be solved. The second thing is that once you find the proteins that are good predictors, that's not a conclusion that from which you can draw causality. That's an inference that at best is a suggestion of what could be at play. But you see, we are embedded in the world of the experimentals. So as soon as an hypothesis is made, it can be tested biochemically. With that corroboration that doesn't come from machine learning, but you know, to tell us out of the ones that we know are good predictors for good reasons, can we shrink the group a little bit? Then, as in normal modeling, you can decide to make a model that is less accurate, but instead of having 10 fields, it has 1 or 2. So you ask me, where do you stop? Well, I stop at 90% agreement. But if you wanted to have 100%, you could try to make models that are even more complex. Of course, as you know very well, that if you have a model with 20 equations, even if each of them was diffusion, each with parameters unknown, the robustness of that model, even if you're under diffusion that is more descriptive, can come to question. Hence my insistence on extracting from those models qualitative mechanism that you can directly test. Also because incidentally, that's what our experimental friends are interested in. They're also interested in prediction for an engineering perspective, bioengineering perspective. But from a scientific one, is that little bits of qualitative knowledge that you can extract from this messy pipeline that they're after. And me too.
So thanks again, Vincenzo. And kind of to follow up on that comment and to perhaps generalize a little bit to some of the other talks we've heard today and talks we heard yesterday, and I suspect some of what will be discussed tomorrow. If you go back to this cartoon of the descriptiveness of the system that you had on the last or second to last slide, this is something that you just referred to in terms of the extraction of qualitative yet meaningful information. Maybe I'll just wait until we get there so that I'll stare at things together. No. Okay, this works too. You had on your almost last slide where you had the experiments. Anyway,
keep talking.
Right. So basically the question is that we often hear about this kind of tension between descriptiveness and interpretability and accuracy. And this has come up in the context of imposing constraints on this area. Okay. I think we've all can think of cases where those constraints might not have limited the accuracy. But I have also seen many cases where it has reduced the complexity of the model in such a way that allows it to be used in more either generalizable ways or more efficient ways. And there are some common metrics for assessing that, number of parameters in your neural network. But extracting what you've said and what some of the other speakers have said, can you comment on, and maybe we have other questions about how to generalize some of those that concept of interpretability in a space where we still want some sort of quantitative interpretation, but the qualitative interpretation is also important.
Yeah. Well, so as I said, since our goal is to write equation, interpretability is always there. Because the equation, you can read them, and they may be wrong, but there's no problem with interpreting them. Our perspective is that whatever we know that happens for sure. For example, geometric things like advection, but we know that every fluid, if it moves, will have that. So that we impose it as a constraint. What we're a little bit more reluctant to do is to say that what's on the right-hand side will be a dynamics that follows from an optimization principle. Because that would, if you have something that minimizes energy, you could have that. But for us, in which all the fields are essentially unconstrained, the only thing that we can put is biological assumptions. But even with that, we're a little bit careful.
On the cartoon, you had physics inspired. Basically, what I wanted that cartoon up on the screen for was you had one box that said physics inspired
Yes.
and one box that said physics constrained.
Yes.
And what I'd ask if you could elaborate on what you think those two boxes mean there.
Yeah.
What those mean in this context, but also how we might think of that distinction that you have drawn literally on the slide more generally in these other contexts.
Yeah, so the extrapolation of the generality beyond, say, using it in this context is something that has to be done with a teacher thought, of course. But in this context, the physics constraint model is when we impose mechanics to hold, and we just enhance it a little bit with information coming from biochemistry. There we assume that the structure exists and we enhance. Something like, for example, the Green's functional neural network that were mentioned before is more agnostic. You assume that there exists a number of fields, that some structural equation is satisfied, maybe inspired by physics guesses, but not what that structure is. And that is what we call physics inspired. I think that personally I consider that the most dangerous of terrain, but it's the one that you do when you know absolutely nothing about it. And I think we're going to hear more this afternoon about how you can do those things meaningfully, but I stress, those are techniques that I would use only when I don't know the equation at all. If what you want to do is to describe turbulence, and you know that there's Navier-Stokes, you don't use that. Okay, because of the others.
Thank you. Okay, I think we have.
Yeah, we have
for another
time.
question.
But lunch is ready whenever.
Can I ask about the data set,
Yes.
which seems incredibly rich?
Yes.
And how much of your conclusions really depend on the resolution of the data or the quality of the data?
Yes.
And can you imagine, what if you have coarser data or finer data, how that might have affected your conclusions?
That's something that we try. I mean, we try to downsample the data, and for our purposes, of course, we have the advantage of the data taken with high precision. It is sometimes interesting to do that deliberately because it's an implicit way to ask questions about coarse grain. And one particularly interesting way of doing that is that, when you look at, the first place where we did that enormously is right here. If you look at this image, we in fact done that, exactly what you asked. Let me tell you, I didn't explain it in detail. When you look at this picture, what you see is a grid, a network that lives on top of the cell. And this network is important because the cell splits and die, and that changes how they interact with each other. But what we've done when we processed these images is to actually blur it to the point that we're now describing this thing that is a discrete distribution of a signal along the junctions into a continuous field that knows the axis of orientation of this junction and the strength of the field. So that's blurring the data like crazy. We've done an implicit coarse grain. Another thing that you can be doing in parallel, we're doing, although I didn't detail it here, is to write down graph neural networks that take into account the full structure of that. And we're in the process of understanding, as we coarse grain our data, or if we lower the resolution of our data, can we merge these two descriptions, which is a poor man way of renormalizing if you do a real space renormalization of the model. That's good.
Okay, if there are no more questions, let's thank Vincenzo again.